my name is Dina Artani and I'm a third-year biological sciences student in Qatar University. And this is a simplification of animal histology 312. But first, what is histology? When was it discovered? How do we use it? Why do we use it? And who uses it? Histology is the study of microscopic structures of tissue. So basically, we study tissues up close and personal. It's achieved through microscopes, different tools, and a series of steps that we will be further discussing later on in this video. Why do we do this? And why is histology so important? Histology is very important and it's used in medical fields, in research, for biological students, biomedical students, almost any student that has to do with identifying human diseases or anything to do with labs will require the skills of histology. It's important and used to identify tissues, their structures, their functions, and if they are diseased or healthy. So when was it discovered? As we previously mentioned, it does require microscopes and proper staining and for the identification of cells and their functions and structures. Therefore, it was pretty recently discovered. It was around the 19th century that people started seeing and staining tissues and viewing them under microscopes. How is this possible? How do we achieve this and view tissues under microscopes? You can't just hold a bone and put it under a microscope and expect to see every cell structure and identify its function. You need to extract a part of that tissue and stain it and go through 7 steps to maintain the exact structure of how it was in your body so you can view it exactly as it was in nature. So to study the cells in a certain desired tissue sample, you need to go through 7 steps that vary and start from extraction of your sample and end with mounting that sample onto your microscope. Now, you need to identify which specific tissue you want to extract, and you also need to know how to use and utilize the tools that you have in your laboratory so you can be able to study the sample that you have extracted. You should also know that this is very important because any errors done in the process of preparing of the sample might damage and alter the results that you view in your sample and in your microscope. So bearing that in mind, let's start with the first step, which is sample extraction. You need to clearly identify which tissue you want to extract your sample from, and you need to extract your sample and it must be smaller than 1 centimeter. And you must also note that if the sample was taken postpartum, as in the tissue that you are extracting from is from a dead organism, then you must rapidly extract your tissue before it starts to deteriorate and it changes how the tissue actually looks and functions in a live body. Second step is fixation, and it's where you fixate your sample into a solution, whether it be alcohol, formaldehyde. Most commonly it is formaldehyde. What are the functions of this step? You fixate it to harden a soft tissue. It prevents deterioration of your sample and it also gives it a higher affinity to the stains that you will be adding later on. The third step is dehydration, and you dehydrate it through putting it in alcohol, and this is one step, but it's actually multiple steps in lab work. You must first put it in a 50-50 water and alcohol solution and then move on from 50 to 100 or 70% alcohol. After the 70% or 100% alcohol, you add it into xylol that replaces the alcohol in the tissue or your sample. It is important that you follow these steps of different solutions and different concentrations of water and alcohol because you don't want to shock your sample and you don't want to shock your tissue. It is filled with water, and if you just put it in 100% alcohol, you will damage the tissue structure and the cell structure and the way they are formed because of the sudden exchange of 100% water to 100% alcohol, and the exchange of these fluids within the tissue happens very rapidly and very strongly that will affect the structure of the cells and how they are organized. The fourth step is embedding. Now, you embed your tissue with paraffin or any embedding agent to make it harder and make it easier to cut and slice your sample to see a very thin layer of your sample. And you basically do this by adding the embedding agent and putting your sample in it. So the embedding agent occupies all the spaces that were occupied by water in your sample. Step 5 is sectioning. And sectioning basically means you're cutting off any excess wax and you're cutting off your sample into very thin slices so that you can put it under the microscope and view it properly. And this is achieved through a machine called the microtome. And the microtome basically just sections or cuts it into very thin slices and makes it about 3 to 10 micrometers. Step 6 is staining. And you can either stain your sample with basophilic or eosinophilic stains. And there are a range of them, and you choose based on your desired sample. You need to remove the paraffin from your sample before you stain because the stains most likely will not stick with paraffin in your sample, so make sure to take out the paraffin and add in water because that's where the stains will be soluble. Then stain your sample. Step 6 is very important because it enhances the contrast of the things in your sample, and it helps you see, for example, a nucleus clearly or the cytoplasm clearly, and it helps you identify the different structures in your sample very easily and clearly. Then we move to step 7, which is the final step where you get to see your section sample tissue. The last step is the 7th step, which is mounting. And mounting has 4 sub-steps. The first is washing off the excess stain of step 6 just to make sure that everything that has a stain on it is stained and everything else does not have any excess stain. The second is that you need to dehydrate your sample, and that is by passing it through different concentrations of alcohol. And then you need to remove and clear the alcohol that you had just added so that the unstained parts of your sample that you will be viewing under the microscope will appear transparent. And the final step is where you need to add the mounting medium, and this is a medium that adds a refracting agent to your sample or your tissue, and you can add the cover slip on top of that, and now you can put your prepared slide onto the microscope, view it under the microscope, and identify the structure or the tissue that you have and its structure. An important thing to remember throughout these videos is prefixes and suffixes. The prefixes and suffixes might sound weird to you because they are all mainly in Latin or Greek, but they're fairly easy to remember. For example, in muscles you have myo or sacro. Anything you hear with myo or sacro belongs to muscle tissues. For example, a myofilament, a sacrolema. These are all structures of a muscle tissue because they start with the muscle prefixes. And every tissue has its own prefixes that identify, and this will be very helpful and help you catch on because as we go along this course, there are many tissues and different structures that we will be discussing, and this will really help you get the hang of it. I hope that this helped you, and I look forward to meeting you all next video.